I'd like to introduce Lori Mignon to the stage. She's going to kick us off with what we're seeing in patients today, so lessons that we're learning in the clinic.
Thank you. I'm really excited to be here with you today to be able to share with you some of the clinical benefits that we've seen across our patients. And so this will be a packed presentation. I'm not able to touch upon everything we've seen, but I'll try to do my best. But we have a lot of families here who have been treated with some of our ASOs, and so feel free to talk to them if they make themselves known to you. But first, I wanted to go back to something that Stan mentioned before. So we really have had a lot more applications that have been submitted to us than we ever expected. And so over the last 5 years, we've had a cumulative number of about 400 applications, or between 300 and 400 applications that were submitted to n-Lorem. We've accepted about 190 patients. So we're working towards treating 190 patients. And like Stan said, this really had forced us to grow much faster as an organization than we ever thought we needed to. Also has forced us to get more fundraising going and fundraise for more patients that we want to treat. But at the same time, this allows us to learn much faster about some of the diseases we're treating because we can treat a lot more patients now. So today, what I will cover in my talk is I'll first give you an overview of the clinical development approach that we're using at n-Lorem so that everybody who may be new understands the process. And then I'll give you some highlights of our clinical experience. And I really want to show you that we're using a modified crossover design. I'll explain to you what that is, but it really has shown us that we can use that design to be able to track disease and track treatment response. I'll hopefully show you that some of our ASOs are really moving beyond just halting a disease, but we've even been able to have patients reach new strides that they didn't reach before. And then we have now been treating patients for multiple years, and so we'll show you that data, and really the benefits are sustained and they're varied across the board. So let's start with the clinical approach. So for somebody who doesn't know or hasn't seen the entire process at n-Lorem, everything starts with an application, and this is really all the steps. And what I will show you is that clinical expertise and experience is really important at every step, and we have different committees that help us throughout. So this all starts with the Access to Treatment Committee. So that's the committee that reviews all the applications that we receive, and they look at the genotype and the phenotype of the patients, making sure that every patient is amenable to receive an ASO medicine and that there is an ASO strategy there to be able to treat these patients. Once an application is accepted, you heard from Stan, the investigator will need whole genome sequencing on the patient, and we need patient cells, iPSCs, fibroblasts, as he said. And then the ASO discovery process can really start once we have the whole genome sequencing and the cells. We do a lot of in vitro tests to find the right ASO, and then we do some in vivo tolerability assessments, and then at that point, we have a research management committee that is made up of n-Lorem people and external people, and we review all the data that we gathered to be able to choose the best ASO to go into a GLP toxicology study. This is the animal study, a repeat dose animal study that is required by the FDA before we can treat any patients. So in parallel to that, once the drug is going into GLP toxicology, we also do manufacturing, sterile fill to make sure that the doctor gets drug in liquid. But on the clinical side, we do a lot of work in parallel. So we work with your physicians to develop the right treatment goals for each patient and to choose the right outcome measures. And so we have another committee that's called the Study Treatment and Assessment Review Committee, who then helps us with the physician to make sure that we have the right outcome measures, that we're looking at the right treatment goals. So after that, once we have all the data together, we have a beautiful protocol put together. We put all the information together for an IND. And the IND is an investigational drug application that needs to go to the FDA. And so at that point, you will have FDA people who will review all the documents that we send. So you can be sure that the FDA has thoroughly reviewed that, and once they give the green light to go, then we have to go to the institution, because the institution has another independent review committee that needs to review the protocols and everything to make sure we're going to treat the patients in an ethical manner. Once we do that, then we can start treating the patient. And at that point, we then have a data safety monitoring board that reviews all of the clinical safety data on a quarterly basis. So my team puts together all that data, sends it to them. It's seriously a ton of data, and we make sure that the patients are monitored and that the drugs are safe. But assessing clinical benefit, I'm going to focus now on benefit. I have a talk about safety later, so right now I'm just talking about benefit, can be hard in nano-rare patients, because nano-rare patients come to us with a unique combination of disease and clinical phenotypes. So a lot of patients have severe phenotypes. They've had disease for a long time, so there's advanced disease in pretty much every patient we treat. And then the patients are on multiple concomitant medications. So we have to take all of that into account when we try to understand the benefit of the ASO that we're giving to them. In addition, for a lot of the diseases that we treated, there's actually a lack of natural history data, because the disease might actually have been discovered 2 or 3 years before, so nobody has had time to really try and understand the trajectory of the disease. And so when we're treating a patient, we don't actually really know what the trajectory of the disease would have been, what we are changing, if we're changing anything, and so we kind of have to think about that all the time. So what we're doing, because if you don't have a lot of natural history data, we are obviously not using placebos in our trials, so you have to use a different trial design. And so we're using the modified crossover trial design. What does that mean? Well, basically, we're comparing pre-treatment data to on-treatment data. And so we do need to start early to work with our investigators to really focus on what is important to the patient, what is it that we want to measure, what's the treatment goal, and what are the outcome measures we're going to use to track that treatment goal so that we can assess that during the time that it takes for us to develop the ASO. So we have some time. So we have the ability to really actually look at what that natural history part is, even if it's just a year or 2 years. And then we can compare that to the on-treatment data. We also know how to interpret clinical data by now in individualized patients. A lot of times, people say you have to look at a population. Well, we don't have populations of patients. We have one patient, and we focus on one patient at a time. But so even though we focus at one patient at a time, we use standardized outcome measures that have been used in other diseases. And so we understand how these measures change in other diseases, and we can try to infer how they're going to change in this patient, or what a change means in this patient. We also try to harmonize the measures across programs and across patients. I mean, like Stan said, we don't really look at disease based on the name. So if somebody has communication issues, we're going to look at communication, and we don't care what genetic mutations they have, and I'll talk about that in a bit. And we want to make sure that we really have high quality data, because obviously, the data will teach us a lot about one patient at a time, about one patient compared to another patient, and at the end of the day, hopefully teach us about health and disease, like Stan said. All right, so let's go into the clinical experience. So if you were here last year, and if you weren't, I'm showing you the numbers of last year and the year before. Last year, we were treating 15 patients across all the organs we can treat with ASO, so CNS, liver, kidney, and the eye, using all routes of administration we can use. And we were at a cumulative doses of 68, and we're very proud of that. This year, we are now treating more than 30 patients, and I stopped giving real numbers because we just dosed 2 more patients last week, and it changes all the time, and I can't keep track, but it is definitely more than 30. Again, still across all the organs, using all route of administration, and we have now given more than 180 drugs across all our patients. And so this is really starting to accumulate, and we start to really understand what's going on. And like Stan said, we've treating more than 90% of all nano-rare patients that are being treated with an individualized ASO. And so we're really trying to gather all that information. And we have had an excellent safety and tolerability profile, and like I said, I'll talk about that in a couple of hours today. So across all our program and across all evaluable patients, we have had about a 90% success rate, we would say. What is an evaluable patient? Well, it's a patient who's been treated long enough for me to be able to look at some data, right? So the cutoff that I made is they have to be treated for at least 6 months, meaning receiving at least 4 doses of ASO, and have had 1 post-baseline visit. And so sometimes some protocols are different. Sometimes our first post-baseline visit is really just at 1 year, because we know it's going to take time to see changes. But really, that's what we call evaluable patients. So out of the more than 30 patients we've been treating, we have 22 evaluable patients that we're going to look at right now. 9 of those are patients with ALS, and I'll go over that in a little bit, and then 13 patients are non-ALS patients. They include 90% patients with CNS diseases, but we also have 1 patient with a retinal disease and then 1 patient with a kidney disease. And so we would say that out of the ALS patients, 5 to 6 out of the 9 patients are showing encouraging clinical benefits that I will show, and Dr. Schneider is going to talk more about that after me. And really, out of the non-ALS patients, actually 13 of 13 are showing encouraging clinical benefits. Obviously, not in every outcome measure, not on every treatment goal, but really giving us encouraging information, for the future. We see benefits across all the organs, and we see benefits early in treatment, because really 6 months is actually pretty early when you think about it, but also at low doses. And so something I didn't really mention before is that for every patient, we do a very slow dose escalation. So we will always start with the lowest dose and then increase in a stepwise manner. Even if another patient has received the same ASO before and we know it's safer at a higher dose, we just want to make sure that in this new patient, the drug is safe. So I grouped the different diseases that we're treating, based on the disease type: neurodegenerative, neurodevelopmental. Some diseases have a developmental onset. Some patients then reach milestones, but then there's a regression, so there's neurodegeneration. We have 2 patients that are treated for dysfunction in the autonomic nervous system, progressive kidney disease, and retinal degeneration. And again, we're really focusing on the phenotypes, and so the way that I will go over this data, especially for the CNS diseases, is kind of focusing on domains. And I'll try to make sure to always let you know which patient was treated, what mutation, so you can understand that better for your own families, but it's really based on the symptoms. So for a kidney disease patient, so this patient was 44 years old who had SAA amyloidosis when he started treatment. 2 of his family members passed away from the disease, so we did have some natural history study actually in the family here, understanding at what age the family members passed away and what happened to them. He had mild to moderate loss of kidney function at study start. And so this graph, just to orient you, it's measuring glomerular filtration rate, so measuring kidney function. The black dots are basically his level of glomerular filtration rate before treatment. The open dots are, if you just took that trajectory and you keep going, right? Because that's the trajectory he was going to. Higher on this graph is better, means normal kidney function. You do not want to get to stage 5 because that's kidney failure, dialysis, and that's what his sister had to go through. After 20 months of treatment, this is what his glomerular filtration looked like. So we did stabilizes, if not slightly going up. But really what that means, obviously it's good for his kidney function. He's not going towards dialysis anymore. But this patient has been able to stop some of the concomitant medications he was taking. He's been able to be more active, to start working out, to just have a better quality of life in general. So moving on to ALS. And again, I'm not going to show much data because Dr. Schneider is going to go into this in more detail. But we've been treating 9 patients with ALS. All these 9 patients have the same mutation in the CHCHD10 gene. And this was their ALS-FRS score before treatment. So the ALS-FRS score is really a clinical measure that looks at motor function and ability to breathe and swallow. So it's really a clinical analysis of kind of like their gestalt. And you can see the trajectory of where he was going. Going down means bad. Just wanted to make that point. So when you look at it, we have a lot of patients where really now the slope is going in a different direction, right? And so that's good. That's not all of it for ALS, because you still have to look at respiratory and bulbar function, but this is already a good sign. All of these patients also showed a reduction in neurofilament light, which is a marker for neuronal degeneration or neural injury. I do want to mention that one patient, not an evaluable patient, but I wouldn't be complete if I didn't mention this. Another patient with CHCHD10 actually passed away, unfortunately. They had a low ALS-FRS score at start. They were already on ventilatory support. overnight at study start, so they were a lot more progressed, and so we probably didn't get to that patient on time. And they succumbed to a respiratory infection that they just caught. But what we have heard from the wife of one of these patients is actually that for them the future is hopeful, and so not a lot of people can say that, with ALS. And again, Dr. Schneider is going to talk more about that. So now I'm going to move on to the neurological diseases, and like I said, I'm just going to focus on symptoms and domains that are common to a lot of patients. So let's focus on seizures first. So we've seen improvement in seizures in multiple patients. In our first patient who got treated with an ASO, our KIF1A patient, we saw a significant reduction in epileptic episode, and this was published last year already in the New England Journal of Medicine. And so you can see on the far left, the dots are high at the top, and then it really decreased over time. In another patient who is treated, who has an SCN2A encephalopathy, we've seen the first, seizure-free days, really, or a lot more seizure-free days. And so if you look at the graph here, the dark green represents the percent of seizure-free days, during that period of time. And so the more dark green, the better. And we had another patient, I didn't put a graph here, who also had a mutation in SCN2A, had to be on extremely high doses of phenytoin or dilantin. And that drug can be very tricky because if you go to really high doses, it can actually become toxic, and so you really kind of want to not have to give that drug. Well, that patient has been able to stop the dilantin completely. And so the ASO is helping. He's on a couple other seizure meds that he was already on before, but really he's been able to remove one of the main medications that was causing some side effects. Now let's move on to communication. We all know a lot of our patients who have neurological diseases will have issues in communicating with, their peers and their family. And what we do is we use the Observer-Rated Communication Ability Scale. This is a caregiver-reported scale. And here what I want you to see is that across different patients with different mutations, we've seen improvements in communication. So to orient you here, bars that are going up, it's a change from baseline, right, in this measure. And you can see that 6 months, 12 months, or 18 months, obviously some patients have been treated longer than others, so that's why I don't have the same amount of data for everybody. But basically going up is positive. And the dotted line at 5 is what actually pharmaceutical companies in bigger clinical trials use as what is clinically meaningful in Angelman's, for example. So a change of 5. And so you can see that we're hitting this and beyond and, getting there for some patients. But again, I don't want to get stuck on the number, but like everybody I've talked to said, communication has actually improved in their patients. Let's move on to mobility and motor control. I think that's obviously also a big one, and that's also a big one that affects a lot of patients with neurological diseases. So I'll show you a couple of videos because we've had 2 patients who were able to take their first independent steps. So they had been able to maybe walk with a walker or with the help of somebody, but their independent step is where it really became important. And so I'll start this video as I talk. And so this patient, this is a video of 4 weeks after their 4th dose. So this is 2 weeks after their 4th dose. They're still holding on to mom, or mom holds on to them. But then you can see 3 weeks after that dose, he can walk by himself towards his dad. And again, obviously not, normal walking, but much better than what he did before. And what was interesting is that at that time of the study, actually, towards the end of the dosing window, he actually would have to hold on to mom again, right? And so we could definitely tell this was the ASO, and the ASO's effect was coming on when it started to go off. And we've been able to now really titrate the dose and work with the physician to make sure that he basically just keeps going. The second video I want to show you is another little girl who's actually here in the room who was able to take her first steps independently as well. And this is a patient who has a neurodevelopmental disorder and due to a mutation in HNRH-PH2. Patient got treated at 8 years of age and has received 5 doses when that video was done. Mom narrates over the video, so I'm just going to shut it and then let you guys listen.
We waited and we prayed and we hoped for the day Lakin would walk. For years, we believed she might take her first steps before the age of 5, but that birthday came and went. Lakin started therapy at 13 months old. We tried everything: walkers, exercisers, endless practice. She could take a short distance with help, but she never could coordinate her movements or find the confidence to let go. Then, after a few ASO treatments, something extraordinary happened. Lakin began to walk. At 8 years old, she took her very first independent steps. 8 years of waiting, 8 years of working, 8 years of hoping, and suddenly it happened. It felt like nothing short of a miracle.
And Lakin is in the back if you just want to give her a high five later. Whew. People, don't make me cry now, come on. It's not okay. All right, moving on. Other movement things. Now I can't even speak anymore. Okay, other improvement in motor activities that have helped patients in daily activities. So for example, we're looking at dyskinesia in some patients. They have a lot of dyskinesia at study start. And so this is a patient, again, with SCN2A who's really seen a decrease in his involuntary abnormal movements. And this was the patient that you saw walking around. Spasticity is also something that happens in a lot of our patients, right? And so this is data from a type 4A patient. So you can see, if you just look at the graph, you just want to focus on the wrists on the very right. So he has had less spasticity in the wrists. So it hasn't reduced necessarily everywhere in every of his limbs, but again, these are steps, baby steps. This is what we see today. Maybe, in another 6 months, we'll see more improvements. And we've also had a couple of patients, again, those that are treated with HNR&PH2, for an HNR&PH2 mutation, that have really seen a reduction in sustained contractions. And so the first patient actually went from having contractions to no contractions now after a year. And again, I think, as you can understand, this really helps in their daily activities just to do anything that they normally do. And we've also heard from a KIF1A mother that for her child, she's noticing lower tension in the legs and a stronger core, not as many collapses when she's crawling around, and better balance overall, and improved fine motor control. So her hands, tremors have gone, and this is really something that we see in those KIF1A patients we're treating, where really the tremor has gone completely. And so excited to see what's coming next. So now let's focus on improvements in autonomic function, something that we didn't really expect with intrathecal injection. So again, in our patient with SCN2A, this patient had a lot of GI issues. And so to try and track this, sometimes we have to use scales that are a little bit rougher because we don't have good measures. So we were using the Bristol stool scale that basically just assesses bowel movements. And so we asked the mother to report on bowel movements. And the Bristol stool scale has a range from diarrhea at 7, constipation at 1, normal in the middle, right? So you really want to be in the green, but this kid was not. This kid was in red. Well, as you can see here, over the time that he got treated, he really has improved the bowel movements, has been able to maybe not have to take as many enemas, and again, just things that really help in your quality of life and in taking care of the children. And then in terms of sensory processing, again, something also that was new for us to look at. Here is the data I'll show you on a patient who has a mutation in ALP1 that causes dysfunction of the autonomic nervous system. And so he had issues detecting temperature change, or detecting temperature, period. So I want to orient you to the slide. This is really showing improvement of detecting cold or warm. So at first, whatever he was touching had to be at 0 degrees for him to realize it was cold. 0 degrees is freezing, so that's really cold. But now, he can sense it at a much, lower cold temperature. And the same for warm. And one of the statements we heard from the physician that kind of stuck with me was that he said, now I can feel heat in my mouth when I'm eating. And it's just something random that I just take for granted that I feel when I burn myself or not. So I think this is really important. In the same patient, what they've also done is to do biopsies at the bottom of the leg and just above the knee to look at nerve fibers, because the nerve fibers are degenerating in this disease. And so just focus on the green fluorescent areas on these two graphs. That's the nerve fibers staining. And so after 9 months of treatment, there was a lot more nerve fibers that were regenerated. And so again, kind of shows that, the effect of the ASO on really the entire disease and different measures. And the father here stated that it's been the best year for his son. So, autistic behaviors, I think everybody knows that this is also one feature or one symptom that really goes across all of diseases that we treat, basically. And again, 2 patients here who really see an improvement in some of their autistic symptoms, for example, irritability, social withdrawal, stereotyped behavior, hyperactivity. Again, the bars going down show that it's getting better. Not every patient reacts on every symptom the same way, but you can see that there's some domains where patients really get better, and so that means that they might not get stuck in one activity or one movement, but they are able to move on and then interact better with their surroundings. And then I want to end this with gained independence, because I think gained independence is really important, but it's really hard to track, right? Because sometimes it's like, well, yeah, they can walk, that's gained independence, but there's so many other things that people do to gain independence, right? Being able to brush your teeth, do your evening routine by yourself. So I have 2 little clips here, and this is our KIF1A patient, and this was the first time that she put on her shoes, and I'll shut up again because it's just exciting how when she calls mom to let her know. So here we go.
You got it.
Like that?
Yeah!
Mommy!
All right, so many exciting things. This little girl now also can walk her dad around New York City in her wheelchair. She just zooms around and he's got to run after her. So I know that Dr. Bain, I think, will show more videos, and it's just very heartwarming to see that. And then here another patient who's been able to focus better and have better motor control. And as her mom was saying, she's improved in various areas. General awareness has improved, her eye contact has improved, and so that eye contact has really also helped in her being able to actually feed herself and go to the right areas when you feed yourself. So again, I think all things that might seem minor to us when we don't think about it, but are really impactful moments in the life of a patient. And then we've also heard from a mother of a child with ATN1 that, he just took off his shirt the day before, and that he hadn't done that for a while. So again, I think small things that are extremely important in daily life. So we do have sustained benefits. We've seen different types of benefits, and they're very meaningful. Like I said, we've shown stabilization in kidney function. We showed stabilization in vision. I didn't talk about that today, but Dr. McCourt will talk about it tomorrow, and you will have actually Grace, the patient, and the mom, Suzanne, here, so please talk to them as well. We've seen slowing or halting of ALS disease, reduction in seizures, reduction in seizure meds. Neuropathic pain is gone. Again, our little KIF1A girl had a lot of pain in her arms and hands and feet, and the fuzzy feeling now has gone. We've seen improvement in motor function, in gait, evidence of ASO on autonomic function, new functions gained, functions recovered. So what we are learning really makes us very excited to be able to treat more patients tomorrow. Again, we don't know if every disease is going to react this way, but we feel that disease-modifying ASOs, if you really work at the cause of the disease, you should be seeing something positive. So we hope that that will be true forever. We've had now multi-year data to confirm long-term safety. Again, I'll go into much more detail on that later. But we see clinical benefits in every organ, early in treatment, and at low doses. So with that, I want to thank everyone. Thanks to our physicians and our staff. Without them, we couldn't treat anyone, so they've done a ton of work. But really, mainly thanks to the families for trusting us and letting us do what we do. Thank you.